All right. So I showed you this slide last class. This is prophase 1 of meiosis, and the reason why I wanted to go over the slide again was one of the main things that is different about meiosis than mitosis is this thing called crossing over. So this is crossing over right here, where one of the main ways you create genetic variation in meiosis is basically the chromosomes of we call these homologous chromosomes. You get one from, let's say that comes from mom, that comes from dad. They're both the same chromosomes, meaning that they, we call them homologous chromosomes. They code for the same traits, but they have different variations. So like, let's say it's like this would be like the gene for eye color here. The version of eye color you get from your mom, the A, T, C's, and G's would be different from the version from dad. And what we do in meiosis is the genetics you get from your parents, from both of your parents, you'll end up doing crossing over where you basically it's like shuffling two different decks of cards together. And what that does is that increases the amount of genetic variation. And we want genetic variation in populations because that's going to give a species the best chance for survival. Okay? It increases the chance that if there's some sort of environmental catastrophe or whatever, there'll be some sort of organism that is better adapted to survive, and so the species line will keep moving on. Anyways, so that's that there. And so what 10.3b is looking at is, well, what is the actual mechanism of crossing over? So how does this work? And the details are actually kind of interesting. So just kind of reviewing, what are we looking at structurally here? Each of the blue lines and the red ones, these are the sister chromatids here. These would be homologous chromosomes. So this, and this diagram, the blue is referring to the paternal chromosome. The red, we'll call it red, is coming from the mother. We talked about cohesins. Cohesins keep the sister chromatids together. So keep sisters together. Then you have the centromere that will also do the same purpose. So they're both kind of collectively keeping the sister chromatids together. So what you do in when you're doing crossing over is you have these DNA breaks at the same locus. Locus is referring to a specific spot on a chromosome. So you're going to break the DNA at the same locus, at at the same gene for each parent. Okay? And the reason why you want to do that is because, as you'll see in the next slide, we're eventually going to cross over. We're going to exchange DNA from the same genes. For example, let's say this DNA break is occurring at gene 5. What you want to do is you want to swap gene 5 from mom with gene 5 from dad. Otherwise, it wouldn't make any sense to swap gene 5 from dad and say gene 7 from mom, because then you would have twice the copies of a gene on one parent and a missing copy of the gene on the other parent. And that wouldn't make any sense. So that's what's going on here. Another thing that's happening as this whole thing is going on is the chromatin is starting to condense even more. Okay, so moving on to the next part. So the unique thing on this slide is you get something called the SynaptoNemo Complex. Easy for me to say. This zipper-like thing that's forming between the two chromosomes. So the purpose of this zipper-like protein complex, the SynaptoNemo Complex, is to attach the homologs to each other. It's basically to bring the homologous chromosome from dad and mom closer together. Kind of like a zipper would zip up a jacket. It's bringing the two ends of the jacket together. Okay. So there you go. So then looking here on the third slide from this picture, here's your SynaptoNemo zipper-like complex, and then this is the crossing over that's happening. And so again, let's pretend we're exchanging gene 5, or maybe it's a collection of genes, gene 5, 6, and 7 are being exchanged. So gene 5, 6, and 7 from mom is being exchanged with gene 5, 6, and 7 from dad. And that then maybe over here, this could be genes 11, 12, and 13 are being exchanged. It's like you're trading Pokemon cards. That's all we're doing, Just trading some Pokemon cards. All right. Any questions about this? You don't really need to know like how exactly this is kind of happening. Just knowing that there's like some of the buzzwords, like the SynaptoNemo Complex is what's holding them together, and then that you exchange different portions. Oh, and then another thing that's also happening here is like the DNA is further condensing and together. And why would the DNA be further condensing? What's the purpose of that? Not space reasons. What do we ultimately end up doing? This is prophase 1. So what does prophase 1 lead into in meiosis? In other words, why would you want the DNA uncoiled, and why would you want it coiled whenever you're going to do meiosis or even mitosis? Why do you want to condense it all together? Whenever you're packing up your laptop and your laptop charger, what do you? You got to wrap it up, right? You got to get it all compact together. Otherwise, it's going to tangle up. You have 23 different pairs of chromosomes. If you don't wrap them up and get them condensed, it's going to be very difficult to separate them during anaphase. All right, and so then this is where you can see we've completed crossing over. Now, once you're done with crossing over, these homologous chromosomes remain, they still remain attached to each other at these structures called the chiasmata. So the chiasmata, this is where crossing over has occurred. Where crossing over occurred. Okay? In other words, it's not some other side of the DNA where they're attached to each other. They remain attached where we did the crossing over, where you exchange the DNA. And then again, the chromosomes will continue to condense because ultimately we're going to end up separating that homologous pair of chromosomes during anaphase 1. Okay. So make sure I said everything. All right, so that's that. So now I want to compare mitosis and meiosis. So looking at them step by step, both of them are going to start with a diploid cell. Okay, so both start with a cell that has 2 copies of all the DNA. They will both go through S phase. So they're both going to go through the cell cycle, go through their checkpoints and whatever. So you have your S phase. And so at the very beginning of prophase, they kind of are looking similar. But what happens in prophase of mitosis versus prophase 1 of meiosis is prophase 1 of meiosis, you get the crossing over happening that we just talked about, right? Where you increase the amount of genetic variation because you're like shuffling the deck of cards. Then looking at metaphase, notice the difference in metaphase. In metaphase, the homologous chromosomes line up separately. Like let's say this is chromosome 1. Like that's the copy of chromosome 1 you get from mom, copy you get from dad. In metaphase, just normal metaphase of mitosis, they line up independently of each other. But in metaphase 1 of meiosis, the homologous chromosomes pair up. Okay, so like chromosome 1 from mom and dad, they're attached to each other by the chiasmata that we formed during crossing over. Okay, so in other words, there's half as many kinetic core microtubules in metaphase 1 as you have in metaphase of mitosis, because you have half as many connections you have to make to separate those chromosomes. All right, so then looking at the kind of the rest of the differences and similarities. So now we're looking at anaphase and telophase. So during anaphase, we separate the homologs, the homologous chromosomes. So notice how we still have the X-shaped chromosomes here. And because we separated the parents, we separated those homologous chromosomes, we are now haploid. That's another key difference. After the first anaphase of meiosis, you go from diploid to haploid. You have half the content of DNA, whereas in mitosis, the ending cells are both diploid. Okay. Now, if you were to count like the number of chromatids, you have like 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. In other words, the DNA content is equal at the end of the first meiosis and the end of mitosis, the DNA content is equal, right? There's 6 chromatids, but the amount of like unique chromosomes is in half. That's why this is haploid and that's diploid. That's an important distinction to make. And it kind of is something that can be not noticed as well. Then in meiosis, we do the second step of meiosis, where we go from haploid down to haploid. But what happens is, what you're doing in the second part of meiosis is you're getting half the DNA content. You're getting half the DNA content because the sister chromatids now are separating. We separate the sister chromatids during, so anaphase 2 of meiosis is similar to the normal anaphase of mitosis. I'll say that again. Anaphase of anaphase 2 is very similar to just your normal anaphase of mitosis, where you're separating the sister chromatids. The difference is, whenever the daughters that come from it, you get 4 that are haploid, as opposed to 2 that are diploid. Okay? And they're going to have half the DNA content. So here there's only 1, 2, 3 chromatids per cell, as opposed to 6 at the end of mitosis. Any questions about that? Okay, here's kind of a chart form showing a lot of that. But there is some differences here. So DNA replication, that's the same, right? They're both occurring during interphase. Okay. Now, what is worth mentioning is that whenever you do that second round of PMAT, you don't do S phase again. Whenever we're doing meiosis 2, we didn't replicate the chromosomes again, because that would defeat the purpose of dividing up the DNA content, right? You want to have gametes that have only 23 chromosomes, so that when you have like the sperm and egg coming together, you get back to the original 46. Number of divisions, you just do 1 in mitosis, but in meiosis you're doing 2. Remember, meiosis, they don't include the second P, they don't include prometaphase, whereas mitosis we do. Synapsis of homologous chromosomes, you don't have that in mitosis, but in prophase 1 of meiosis, that's where we have the crossing over, so you get some genetic variation. Number of daughter cells and their composition, you get 2 identical daughter cells, whereas in meiosis you get 4 haploid that are different from each other. Okay? And then the last, actually that's it. It's really the main differences. So yeah, here's everything just kind of in words. Let me make sure I said everything. Another way of saying like the daughter cells have the same number of chromosomes as the parent cells is we conserve the number of chromosomes, meaning diploid parent, you get a diploid daughter, 46 chromosomes in the parent, 46 in the daughter. Whereas in meiosis, we reduce the number, right? You go from diploid to haploid, or you go from 2 copies of all chromosomes to only 1 copy. Or at the end, you have 23 chromosomes, whereas at the beginning, you had 46. Another thing that's unique to meiosis, and I mentioned this last class, and there'll be a good picture of it a little later on, so I'll just only briefly mention it here, is the alignment of homologous chromosome, of the homologous pairs. They align independent of each other. Called an independent assortment. I'll show you a picture of that later on, so I won't dwell on it here, but just kind of keep in mind that that does exist. You still have the sister chromatid cohesion. You still have those cohesions being broken up whenever you want to separate them during anaphase. So in meiosis, you're separating, again, the homologs in anaphase 1. Anaphase 2, you separate the sister chromatids. Okay. All right, so looking at 10.4. 10.4 is focused on, like, how exactly do we produce genetic variation in meiosis? And a lot of this we've talked about, Although what we haven't talked about is mutations. So ultimately, the original source of genetic diversity comes from mutations. The idea of a mutation is, let's say you have like a DNA sequence like A, T, A, C, G, G, A. A mutation would be like, an example could be like A, T, and instead of there being A there, I got a mutation, and I then got a C right here. That's a mutation. You get a change in the DNA sequence, and DNA ultimately codes for proteins. So what happens is by changing the DNA sequence, you're going to eventually change the protein that comes from that gene. Okay? And most of the time when you get a mutation, it's they call it a deleterious mutation. It's a mutation that doesn't help the organism. It usually gives you a broken protein. Every so often, that mutation might create a beneficial adaptation, like some sort of protein that works better than the original one. Anyways, the point of all that is we talked about crossing over, where we shuffle those genes. Mutation gives us the variation to do the shuffling. In other words, if you don't have mutations, it would be like shuffling like two decks that have the exact same cards. Like, it wouldn't really do much for you. Whereas mutations give us a difference in the deck of cards, so that when you combine them and you exchange different portions of them, you will actually get two different decks of cards. There you go. So there's three things that are going to give you genetic variation in meiosis. I mentioned two of them last class: independent assortment and crossing over. The third is random fertilization. The idea of random fertilization is any sperm, why does that look so weird? Could fertilize any egg. Fertilize any egg. It's just random. There's the sperm and the eggs aren't like selecting each other like, ooh, you seem like I'd like to be with you. It's just kind of the sperm, any one sperm happened to meet up with any one egg. Whenever that female was ovulating that month, she just happened to release that egg. There isn't any the eggs that females have aren't labeled all right, this one's gonna come out next and this one. There's no difference in the eggs females are releasing at one stage of life versus the other. Same with sperm for males, right? Males aren't making super sperm once they're in their whatever age or something. It's just the sperm are the sperm, the eggs are the eggs. They randomly come together. Okay? All right.
That's a crazy call out.
Anyway, so this slide is showing independent assortment. So independent assortment. So we talked about this last class. I just want to kind of establish, make sure we're on the same page with what this is. So I said the idea of independent assortment was whenever I'm showing you these chromosomes lining up, here, you're oh, okay, blue's here, goes blue, red, but it just as easily could have been blue, red, red, blue. Or it could have been both reds on one side and both blues on the other side. It's random, right? For humans, we have 23 chromosomes. So if you look at basic probability, let's say I flip a coin. I have some coins somewhere. Here we go. If I have a coin, and this coin, there's two probabilities. It could be heads or tails. Two probabilities. What's the odds that if I flip this coin, it's gonna land heads 2 times in a row?
25%.
25%. The exponent here is the number of times you get it correctly. So in other words, it would be the number of possible combinations. So if I flip the coin 2 times, 2 possible variations, and I'm doing it 2 times, that would be 2 squared is 4. There are 4 possible combinations. For humans, where the 2 is coming from is this blue could be here, or it could have been here. It could have been on the left side or the right side. It's heads, tails. Because we have 23 different chromosomes, the amount of variation that independent assortment gives us is 2 to the power of 23. So like that's the number of, which is a giant number. I don't even know what that is. Like it's like a gigazillion or something, you know. Anyways, their independent assortment by itself gives us a ton of variation. Is the point? All right. 8 million, more than 8 million combinations is what it would be. Okay? If you were to go across all your chromosomes, that's kind of wild. But that's not just like, that isn't there was 3 forms of like variation. That was just one of them. So then if we look at crossing over is our second source of variation. So like at crossing over, so you have 23 pairs of chromosomes, right? There's like usually 2 to 3 sites of crossing over for each of the 23 different chromosomes. In other words, we will do crossing over maybe like up to 3 times 23. Like, is that 16? There's like 69 different times you could do crossing over during meiosis. 46 to 69 different events of crossing over during meiosis. So that would add even more variation on top of independent assortment. So there's that. And then, okay, and then, that's not what I want. And then you have random fertilization. So again, random fertilization is any sperm confused with any egg. Okay? So that, if you were to combine like, what are the odds of like, you have the fusion of any one sperm with any one egg? They tell me it's something like, if you combine so if you combine independent assortment, so you have 2 to the 23rd different possibilities of how the chromosomes align, which we said was 8.4 million possible combinations, and you combine the fact that any sperm could fertilize any egg, you would get to 70 trillion diploid combinations. 70 trillion combinations of kids your parents could have had. And that isn't all of them because these numbers aren't including crossing over. So like, I don't even know what is even beyond trillion. What's it? Quadrillion?
Yeah.
Like I think there's like in the quadrillions of possible combinations of like babies to like a couple could have, which I think is kind of wild. So you are unique, right? Anyways, that's what they're trying to say here, right? Crossing over adds even more variation that's not accounted in that 70 trillion combination number. Okay. All right, and again, the purpose of all that genetic variation is natural selection works best when you have a lot of different variation, right? If you have a lot of variation, then you're going to increase the chances that one of those organisms will have the right variation to survive in the environment best and keep the species line going. And again, don't forget that the original source of genetic variation is typically going to be mutations. I think that's it for 10.4, yeah. All right, we'll take a little break.